The supermarket mango is a genetic clone. If a disease targets that exact genetic sequence, the global supply could collapse into dust. The ancestors of this fruit do not come from manicured orchards. They evolved in the mist-shrouded, flooded rainforests of Malaysia, a massive botanical region encompassing Indonesia and Malaysia. Within these ancient canopies are over 200 wild mango relatives. They produce fruits that look and taste completely different from the sweet, golden varieties we cultivate. These wild variants are a record of how the genus survived millions of years of shifting climates and local pathogens. They represent a genetic library that is critical to the future of our food supply. This grid showcases the physical diversity of Indonesian mango cultivars. The fruits range from tiny green berries to massive, oddly shaped spheres in various colors. This vertical view captures a wild Mangifera quadrifida. Unlike the small trees in commercial orchards, these are towering 100-foot emergent giants of the primary rainforest. Since 1941, Indonesia has maintained a sanctuary for these trees, called the Cukur Gondang Gardens. This single reserve currently holds 208 native cultivars. Cataloging these giants is a race against time. The primary habitats where these ancient trees evolved are being converted to industrial use at an accelerating rate. For decades, botanists categorized these trees using morphological markers, identifying species by traits like the shape of a leaf or the curve of a petal. But in a dense jungle, physical appearances can be deceptive. This animation shows the modern molecular tracking system researchers use to identify these trees. Botanists extract DNA and deploy RAPID markers. RAPID stands for Random Amplified Polymorphic DNA, a technique that uses short sequences called primers to attach to and highlight specific segments of the genetic code. By tracking these segments, scientists organize the mangoes into a branching family tree. They discovered a 31% range of genetic variability hidden beneath physical appearances that often looked nearly identical. The evolutionary value of these trees is not found in the fruit they produce today, but in the specific genetic traits recorded within their DNA. Consider the sweet, golden commercial fruit, Mangifera indica. Now compare it to its wild cousin, Mangifera quadrifida, locally known as the Asam Kembang. This photograph shows the branching inflorescence of the Asam Kembang. It builds its flowers in strict sets of 4 petals and 4 sepals. This image contrasts the mature Asam Kembang drupes with standard green variants. The fruit has a leathery, almost completely black exterior. This cross-section reveals what is inside. The Asam Kembang contains pale yellow, highly fibrous flesh dripping with watery juice, clinging to a coarse central seed. The flavor profile is a departure from commercial mangoes. It combines an intense acidity with a prune-like sweetness, used by locals for traditional culinary condiments. This shows the habitat of the Asam Kembang. It survives as an emergent tree in inundated wetlands and flooded riparian zones of the dipterock forest. The Asam Kembang is a specialized survivor. It has adapted to thrive in waterlogged environments, where standard commercial trees would quickly rot and die. The hearts of mango diversity, Sumatra and Borneo, are experiencing the highest rates of forest loss in Indonesia. This aerial view captures a freshly cleared red dirt logging site. Between 1990 and 2010, Sumatra lost 70% of its forested area. This image shows what replaces ancient ecosystems. Industrial agriculture installs uniform expanses of palm oil monocultures, replacing thousands of species with repeating rows of a single crop. This map of Sumatra illustrates the decline. Historical records once documented 34 wild mango species on the island. Recent explorations found just 12 remaining. Clearing these forests removes an entire biological archive. It deletes millions of years of evolutionary adaptations built into the DNA of the plants that live there. Commercial mangoes lack genetic variation, making them vulnerable to diseases like anthracnose, a fungal infection that thrives in wet climates and destroys orchard harvests. This visualization shows how wild species survive. Trees like Mangifera torquenda and Mangifera quadrifida possess natural resistance needed to block these pathogens, even in the wet climates that kill commercial crops. This microscopic simulation demonstrates the goal of current research: By mapping these sequences and crossbreeding them into commercial lines, scientists can stabilize the genetic weaknesses of our food supply. The surviving rainforests of Southeast Asia function as an insurance policy. The genetic variability found in these wild populations allows for the development of crops capable of withstanding shifting global environments. Preserving these wild fruits is a pragmatic necessity. We are keeping the original genetic material of the genus intact, so it can be used to stabilize the future of tropical agriculture.